Thanks for coming, everyone. This is the fifth and final session in our virtual workshop series on student activities for exploring statistical concepts. Today's topic is comparing means among more than two groups. Our presenter will be Todd Swanson, who has taught for many years at Hope College and is a textbook author and co-author and has won multiple teaching awards in his career. And Todd just retired a year and a few months ago now. But fortunately for us, he's still actively engaged in some projects, including this one. And Todd will be leading you through a student activity for comparing more than two means. So thanks for coming, everyone. I'll turn it over to Todd.
All right. Yeah. So thanks for showing up and attending on this Friday afternoon. And my screen you can see it and all?
Yes.
Okay. Yeah. So the first thing I wanted to talk about was that EA Post website. And I think Alan was going to put that in the chat. But on there, not only my presentations, but our previous four ones, we've got PowerPoint slides, student exploration, solutions to the exploration, links to the applets. And let me just click on here so you can just see it. And I'm going to tell you sort of what I'm going to do and not do. So I'm going to click on the workshops, and here's our group right here. And here's mine on materials. And so the recording will be up there this weekend sometime, but my slides are there. They said a student handout. I'm going to click on that and kind of show you the length of it and the verbiage of it. As I go through my presentations, I'm not going through this at all number by number. I kind of am doing my own thing in a different order and adding some things that aren't on here. But it's kind of a complete module for students to do. So let me get back to my slides. And I want to do a little background before I jump into the exploration, to kind of tell you sort of where we are in the semester when I would be doing this in class, and some things that the students would know and maybe some things that they wouldn't know at that point. So this comes late in the course. I'm comparing multiple means. I'm going to do ANOVA here in a bit. So it comes like in our chapter 9 out of 10 chapters. So late in the course, they've already done inference for a single variable, both proportion and mean. They've compared to groups, proportions, means, match pairs that Jill did last time, and compared more than two proportions. And the students would have seen the simple statistic we're going to use. It's going to be a mean group diff statistic. They would have seen that or comparing multiple proportions. They would have seen this simulation-based inference process that we're going to do many, many times over and over again. So it should be kind of almost boring at this point. Oh, we've seen this before. And they've seen that transition that we're going from simulation to theory-based methods, again, over and over and over again, right from chapter 1, they've done that. And then we got this 3S strategy and the 6 steps. So the 3S strategy is what we're going to use to talk about how we do the simulation, to do that simulation-based inference. They would have seen that, again, over and over again from chapter 1. And that 6 steps of the inferential process, we actually have a chapter before chapter 1 called preliminaries, and that they start that back in that. So again, they've seen that over and over again, but you'll see those things in my presentation. So that mean group diff statistics that I talked about. So when we're testing multiple proportions, as I said, or multiple means like I'm going to do here, we use this mean group diff statistic. That's a very intuitive statistic, one that you can sort of get students through a couple of questions to probably figure out, oh, we could do this. We could do this to find out how far our means are apart or how far our proportions are apart from each other. So it's a very intuitive statistic. And then we kind of segue there to a chi-squared or an F statistic like we'll do today. And here is that mean group diff statistic. It's just the absolute value of the differences between each group, whether you're talking about proportions for those groups or means for those groups. So here I have, this is probably an example we had from proportions. So we end up with these 3 proportions. And basically we want one statistic, one number that tells us how spread out are these numbers. So the first thing you do is find the differences. I've got those in red there, and then just average them. And just make sure you have the absolute values of the differences or make sure they're positive, subtracting in that way. So we're just averaging those differences. And I always think of it as the average distance, because distance is a positive number. The average distance between our three proportions or three means, or today we'll have four proportions. I just had a cat jump up on my lap. I'm to put down. So again, that mean group diff statistic is that average distance between three proportions. And so at the bottom of this slide, say I have proportions of 0.3, 0.4, and 0.9. What is that mean group diff statistic? I gave you nice numbers. This is exactly what I do with my students. So put it into chat what you think the mean group diff statistic is for those numbers. Yeah, I'm seeing a couple of 0.4s. Yeah, I think it is 0.4. So again, this next slide, you've got my three proportions there. Find the distance between those three, 0.1, 0.5, 0.6, add them up and divide by 3. So again, we're finding that number. And also, we would have talked about this when we're comparing multiple proportions, what would my proportions be if my mean group diff statistic was 0? What kind of proportions would I have that would get a mean group diff statistic of 0? Toss that into chat. Yeah, I see a couple all the same proportion. Yeah, they're all the same. There's no distance between them whatsoever. And the bigger that mean group diff statistic is, the more spread out our proportions are, or what we're going to talk about today, our means are. This is also the place where I would talk about follow-up tests. So when we would have done the proportions, multiple proportion sections, we would have done this mean group diff statistic. We would have done a chi-square statistic. And if we got a large enough chi-square that we had strong evidence that there is some difference there, we would have find out, well, where is that difference? And what we do is we do confidence intervals, find pairwise confidence intervals between all of our proportions to find out exactly where that is. And my question I always ask the students at this point is, that's where we ended up finding those pairwise confidence intervals. Why didn't we just start there and do that? And as you probably know, that type 1 error thing. So what I do is go through this following example to kind of introduce that idea of multiple tests and controlling for a type 1 error. And numbers are complete. Well, these numbers aren't made up here, but I'm thinking of them as like made up numbers, though I don't really have numbers here. But I had one student one semester who every day when he came to class had this Rockstar 2 times energy drink in class to kind of pull him through, I guess. So I thought, well, you could do something like this. We do group projects, and students could do a group project like this, compare caffeine effects on things, So I could say comparing caffeine's effects on increasing people's pulses or heart rates. And I looked up and I found Mountain Dew for 12 ounces had 55 milligrams of caffeine. And Monster energy drink had about twice that. So the size of the can is not necessarily the size of the can. I'm trying to make the height like the amount of caffeine. So it's got 120. And then the Rockstar 2 times energy drink is about twice that, 250 milligrams per 12 ounces. So say you randomly assign students to, some had the Mountain Dew, some had the Monster, some had the Rockstar. And you took their pulses before, took them pulses after, and you looked at that mean, that increase in pulse rate. Well, if we're doing multiple tests, of course, we've got 3 means. And if we're pairing them up, like we would have ended up with, we'd have 3 tests we'd have to do. A versus B, B versus C, A versus C. Those 3 won. And if we're testing at the 5% significance level, then there's this 5% chance of making a type 1 error. Remember, type 1 error is rejecting a true null hypothesis. We like to call that a false alarm. You're rejecting the null, so you're concluding something. You're saying, oh, something, there's an effect here, when in fact there isn't. Well, that's a false alarm. So if you're doing this over and over again, it builds up. These type 1 errors accumulate. And if you're doing 3 tests, the probability of making at least one type 1 error is 14%. And doing 4 tests, there's going to be, or sorry, 4 means, it's going to be 6 tests. That jumps up to 26%. 5 means that, or that 10 test, it jumps up to 40%. So that's why we don't end up here. We kind of control for that at the beginning, testing overall test at this 5% level. And then if we find something, then we can look where it goes. Sometimes I go through how these are calculated. It seems like I tended to do that more earlier on and not so much later. I do talk about the 3, the 6, the 10 to try to guide them. Do you know where those numbers come from? And usually through a couple of questions, I can get them like, oh, Pascal's triangle, binomial distribution. Some of them had seen that before. All right, 6 steps of statistical investigation. Ask a research question. Design a study and collect the data. And then explore the data. Look at means, look at proportion, look at graphs. And then you do the inference. And that's the test of significance or the estimation, doing confidence intervals. Then formulate conclusions. That's the scope of interest. Where can you generalize the results to? Can you determine cause and effect there? And then the 6th step, look back and look ahead. Look back at what you did. Did everything seem to go okay? Were there any problems? And then look ahead, like what would you do next? And our students do projects at the end of the semester. And that 6th step, that 6th step there, look back and look ahead, is always important because there are students in an intro class. Something is not going to go right. And that's fine. But I want them to look back and say, well, this didn't go quite right here. And this is how I would do it differently later on. That's an important thing to do. And it's nice when the papers have that in there as well. But you'll see these 6 steps as I go through my module. So here it is, exercise in brain volume. So step 1, state that research question or ask the research question. Brain size usually shrinks as we age. And the question is, such shrinkage may be linked to dementia. But the question is, can we do something to protect against that? Is it exercise? Is it social interaction? What kinds of things will help that? So specifically, they were looking at what types of exercise or activity help prevent brain shrinkage in older adults. This was a paper written 12 years ago. A lot of these people were from China. So the study was done in China, but the lead author was from University of South Florida, I think. And so they randomly assigned non-dementia subjects into one of 4 groups. So the activities are doing Tai Chi, walking. They had a social interaction group, which I'll talk about more in a bit, or none. So they had a control there. And this is an interesting experiment because you hardly ever see this. The study started out with a random sample. This is going to be an experiment, but they started out with a random sample of 250 residents, 125 men, 125 women, age 60 to 79. They had a government list of a specific neighborhood in Shanghai, China. And then those potential subjects, those 250, were visited to determine their willingness to participate and if they fit into these inclusion criteria. So they didn't want any history of stroke, Parkinson's disease, or any other current neurological diseases. They had to have the ability to walk at least 2 kilometers because some might be assigned to the walking group. They had to be able to maintain balance. I think that was just like standing still for 10 seconds. They had to have a cognitive score high enough to rule out dementia. They need to be cardiovascular and muscular strong enough, healthy enough for any of those interventions they might be assigned to. And they did not want them currently participating in any vigorous exercise or currently participating in Tai Chi because they wanted that to be an effect. So if they got in the Tai Chi group, oh, it was something new. indifferent. Or if they got in the walking group, oh, they didn't walk much and it was new. So of those 250 participants, only 51 men, remember they started with 125 men. So 51 men and 97 women agreed to participate and met all those criteria. 11 dropped out before the study began. And then 17 were randomly dropped out. So they had this sample size of 120. So they're going to put 30 in each group. But as things go, 13 dropped out during the study. So they didn't end up with that. They had 107 at the end. So except for the group with no intervention, each group met for 3 hours a week for 40 weeks to their assigned activity. The Tai Chi group was led by a Tai Chi master. The walking group were walking around a track together. The social interaction group met at a community center, and they said it discussed topics of interest to them. And what's interesting about this one is that group stayed together after that 40 weeks. They liked it so much, they stayed with it. I might be hinting at some result here later on. And then there was a control group, and they just received 4 phone calls during that 40 weeks. All right, the paper had this table in it. This is a table not necessarily this one, but I like to show students at least one of these tables. It's like random assignment at work, and a lot of papers do put this out. So you have these 30 people assigned to each group in Tai Chi, walking, social, and no intervention. And what you want is things to be the same across those 4 groups. You want the gender percentage to be the same, the mean age to be the same, the education to be the same. And you've got some things with health things, time spent exercising, smoking, brain stuff, dementia, verbal memory score, Stroop color score, and all. You want that to be all the same. And one thing I typically leave out for my students is p-values over here, because that's always a question I give them. What do we want those p-values to be? We're looking for small p-values? Like we all tend to be looking for small p-values or large p-values. Well, here's the case, of course, we're looking for large p-values. We want no evidence that there's a difference. We want things to be the same. So we should end up with large p-values. And they do that for the most part. There's some that are getting kind of low here, but for most of them are pretty large. All right, still on that designing a study and collecting the data. So to measure the response, the participants had an MRI determine brain volume before that 40 weeks started and then after. And what they're doing is measuring the percent increase or decrease during that 40-week time. So positive numbers are going to indicate an increase, negative numbers a decrease. So what's the explanatory variable? And then what type is it? Toss that into chat so I know you're still alive. What's the explanatory variable and the type, quantitative or categorical? I see Jill got the activity group and categorical. Jennifer, you're an overachiever. You gave me the explanatory variable type and the response variable type. See in my next question. Yeah. Yeah. So I was just describing basically the response variable, but yeah, that explanatory variable is which group they were assigned to, that activity group, and it's categorical. And the response, that's that brain volume change, and that's quantitative. All right, here are hypotheses. So lots of ways you could write out these hypotheses. When you're dealing with 2 variables, you can always talk about there's no association between whatever your 2 variables are for the null. But it's nice to specifically talk about, well, how are you looking at that no association? So in this case, those long-term average brain change volumes, they'd be the same in all 4 groups. And the alternative, at least one is different. Again, there is an association. At least one of them is different. And now step 3, finally, explore the data. So what I like to do before we see things broken up into groups is let's look at the response variable just as that single variable to start get a feeling for that before we break it up. And so here's that brain change percentages. And as I said, negatives, that's decreasing, that's bad. We don't want that. Positive, that's good, that brain volume is increasing. There's all the data combined. So the mean's not much more than 0.138%. And there's the standard deviation about 1.1. And so now when we break this up into groups, I like to think we're adding another variable to this mix. And that sort of gets that idea of, we could add another one after that. If we had one, we could add another one after that, getting into this multivariable way of thinking. Let's toss in a variable, see if that helps explain some of the variability, see if that seems to have an effect. So now got a little complicated graph here, but here are the 4 groups, and I've got the means are here in these red arrows, and they're also over here on the side, and the standard deviation is kind of shown by the width of that red line from the mean out to the edge. So again, 2 questions I'm asking at this point to the students is, did that adding that variable of treatment, did that explain some of the variability? Or also, does that seem to have an effect on the response variable? And so it's not huge, but it is sort of clear when you look at the graphs, you can see there is a difference. And I've got the means highlighted there, so you can see 2 means are here and they're positive, 0.4, 0.4, and 2 are down here, and those are both negative, negative 0.2 and a negative 0.15. And then looking at variability, got 2 of the standard deviations highlighted here because the standard deviation of all the data was 1.1 something or other. And these standard deviations decreased a bit, especially the one for the social group. But the other one's not so much. So if we look at the control group, it didn't. The Tai Chi group, it didn't. Or sorry, Tai Chi group. The walking group, it didn't either. And so we're not really having an effect here of increasing brain volume. They're negative and certainly didn't have an effect in reducing variability as well. And then another thing I talk about with the students here at this point is R squared. So I would have talked about R squared when we compared 2 means. So R squared is just looking at the percentage of variability that's explained by whatever the explanatory variable is. And here we can see 8.3% of the variability in all those brain change sizes. If I go back here, 8.3% of all this variability going back and forth here is explained by which group they were assigned to, is explained by that treatment. And so it's not huge. It's fairly small, but it will end up being significant. And so that's the same idea when you're doing regression or if you're comparing 2 means. You can explain it here. And what I found is, when you're doing regression, at least when we did regression, it was chapter 10. R squared would have been about the last day or the second to last day of class. They never did well on a question on R squared on the final. Never. It was horrible. Now, it's the 3rd time they've seen it. Now they did fine. I should say now. I'm retired, but last time they did fine on it. So I think it really helps to have them see it a few times. All right, still on that 3rd step of exploring the data, we need a statistic, and that's that mean group diff statistic. We had our 4 means here. They kind of look like proportions, but they're all means, these 4 means, and there's 6 ways we can pair them up. So I've got my chart, I've got these 6 differences and find the average of those differences. It's about 0.45 or 0.458. Remember, this is just the average distance between these 4 proportions. And it might have been a good idea. When I did the 0.3, 0.4, 0.9, those are my numbers, we sort of had them almost like on a number line and spread out. Here, I don't have that at all. And visually, I think it's nice to see from low to high and sort of spread out because you're just trying to get at that average distance. How far are they apart? That's what you're measuring. All right, finally, step 4, make inferences beyond the data. So here's where we're doing the inference with this. So again, this is a question that would have asked over and over and over again throughout the semester. What's the likelihood of getting a statistic as large or larger than we got, that we found, our observed statistic of 0.458, just by chance, basically if the null were true, if there's no difference in the brain volume change between those 4 groups in the long run? And then, of course, that's going to be the p-values that are going to tell us that one. And then what types of values of this statistic will give evidence against the null. So get them thinking that, oh yeah, larger mean group diff statistics are going to give evidence against the null. Smaller ones won't. Same thing with the chi-squared, same thing with the F statistic. It's all the same. All right, here's that 3S strategy for measuring strength of evidence. Well, first one, compute a statistic. That was our 0.458. Compute a statistic from the data. Could be a difference in means, difference in proportions, single proportion, whatever the case. Right now, it's our mean group diff statistic. And then 2 is simulate. Identify this by chance alone explanation, and then repeat values of the statistic could happen if the null were true or that chance model is true. If the group treatment didn't matter at all, what sort of statistics would we get? And then we're going to get a whole distribution of these statistics. And then the strength of evidence puts those first 2 steps together. It says, well, where is our statistic in those simulated ones? Is this like something that could happen very easily? Oh, yeah, it happened all the time. Or is it something, oh, that's very unlikely? Or that never happened? Or it's kind of unlikely. Where is that? So we're going to see this process visually, but remember I had 107 in the final sample size. We're going to see it with a smaller data set because I'm going to have some animations going on here. But the numbers are about the same. So again, going through that 3S strategy, statistic, compute the statistic from the observed data. So you had these 107, or I forget how many I have here. I think there's, I forget. I think there's 40 here. We'll see here. So you've got these 40 people. They're signed up to be in this group. So they randomly assign them to one of those 4 groups. You've got the Nun, the social, and the Tai Chi, and they're in those groups. Then they go through that process. They go through their treatments, whatever it is. Their brain volume is calculated at the beginning, it's calculated at the end, and then that response is that brain volume change. So they all get a number in their brains there, tattooed. So that's getting the statistic. We first calculate the 4 different means, and then get that mean group diff statistic from that. So that's that randomized process. So what we're going to do in simulation is basically redo this, but now with those actual numbers. So the second S, simulate, repeatedly simulate values of the statistic that could have happened if that chance model is true. So what's that saying is these group titles, Nun, social, walking, Tai Chi, they don't matter. The null says they don't matter. People are going to have a brain change of, like this first one, of a negative 1.35, regardless of which group that person was assigned to. So let's just shuffle them up, put them in those 4 groups, recalculate the 4 means, recalculate the mean group diff statistic, and this is a value that could have happened if the null were true. If the Nun, social, walking, Tai Chi didn't matter. And you just keep doing this. Do it again. Scramble them up. Recalculate the means. Recalculate the mean group diff. Do it again. And students in my class would have seen these kind of slides many times up to this point in chapter 9. And kind of keep reminding them, we're looking for where is our statistic in there, that 0.45. It didn't happen at all yet in my simulation. So let's do that a few more times to start to see this null distribution getting built up. And again, these are statistics that could have happened if the null were true, that could have happened if that group activity did not matter. And that third S, the strength of evidence, well, where does our statistic fit in there? Right now, 0.45, right about in there. And I can see I've got 5 out of the 30 total. So for my estimated p-value, 5 out of 30, approximately 0.17. So that's the process. And as I said, they would have seen this process many, many times. All right, so let's look at the real data now in the multiple means applet. And I'm going to click on here, but can somebody, like Jill or Alan, put this into chat? If you want, anybody wants to play along with me. So what I'm going to do here is I'm going to click on this applet, and the multiple means is going to be under the quantitative column. And then I'm going to click on that multiple means, then I'm going to get the data, and it's going to be a pull down menu to brain. And then we're going to click on use data. It's the process. So open up the applet under the quantitative response, multiple means, and then here, pull down this, and brain is the first one. So when you click on brain, it loads the data in here, but to actually use the data and see it over here, you have to click on use data. So you've got the data here on the left side of the screen, all the data together before it's split up, kind of like I showed you. And then in the groups, you can add box plots to this. You can make this bigger, oops, if you want, so you can see it. You can make it bigger this way. But there's the data. And then below there, you've got the sample sizes, the 4 means, the 4 standard deviations. And then the default statistic is that mean group diff statistic that we calculated. And it's right here. I'm trying to think of anything else I want to say on this side. So again, like Jill mentioned last week, these applets, on the left side is just the descriptive statistics. We're not doing any inference yet, at least simulation-based inference. And then on the right side is the simulation. So when you click on show shuffle options, this is where you're going to do the simulation. You can do it with data or plot. Plot's going to be the nice one here to visually see things. So when you're clicking on plot, I'll make this a little bit bigger, you see the same data set. Let me get rid of the box plots. See the same data set that's over here. But when I shuffle them, it's going to get shuffled. All of those 107 are going to come together and randomly put them in groups of those same sample sizes, 24, 27, 27, 29, and recalculate all this stuff. So let's see it do it once here. And again, here's the summary of that, the shuffle data. We have a mean group diff statistic that's 0.37, a little bit smaller than our observed. And that point was plotted over here. So just like in the PowerPoint, it's doing that same thing. And do it again. Get a different mean group diff. Close to the same that we had, but a little bit different. And again, and you can build this up slowly or as fast as you like. So here's 5 of them here. And then I'm going to change this to 95. So I'll do 95 more. So I have 100 total. And at some point in here, you're going to want to stop and say, well, where does our observed statistic fit into this? Remember, it was 0.448. So 0.448 looks like right in here. So maybe 2 out of the 100. I don't know about that one. Let's see here. 0.448. Yeah, it's just those 2 out of 100. And I always like to do this before I click on anything, is just to, I'd go up to my screen in front of the room and go, here's our statistic. Where does it fit in here? What's the p-value look like it's going to be? Big or small? Here it looks like it's going to be small. So I've got 100 of them. So let's do 900 more. So I have at least 1,000. And yeah, the p-value looks like it's going to be fairly small. We can build this up till you're satisfied that, yeah, it looks like it's around 0.03 on there. So let me pause here. Does anybody have any questions on the applet and what I've done so far? You can ask it in the chat or you can unmute. Okay, so again, we went through that 3S process, getting the statistic, doing a bunch of simulations of statistics that could have happened if the null were true, if there's no difference in brain change with those different treatments. And then the strength of evidence, where does our statistics fit in there? It's fairly unlikely. Not very, very unlikely, but it's fairly unlikely with that. All right, we're going to come back to this applet when we do the theory-based stuff. For now, let's move on to step 5, formulate some conclusions. We've got that small p-value, so we've got strong evidence that at least one of those long-run mean brain changes is different. And I was asking my students, can you tell which one or which ones? Let's go back to the data and look at it. So we're going to do follow-up tests when we do theory-based stuff. And I always can't help myself when we do simulation. But you can go back to I just go back to the data. Maybe I should have gone back to the applet. Back to the data right here. Kind of like I was pointing out to you, there's 2 that are positive in the 0.4 range and 2 that were negative. So my guess is the social and Tai Chi looks like those are going to be significantly different, maybe from those 2 are negative, because things seem close on the negative and the positive side. They're kind of grouped that way. But we'll see that in a bit. All right. Yeah, as I said, we're going to do pairwise confidence intervals when we go back to look at theory-based. All right, still under step 5, formulate conclusions. So I always ask these questions, can we determine causation and generalization? So you're comfortable with concluding that that the activity caused that difference? Into what populations are you comfortable generalizing the results? So it was a randomized experiment. So very easy. Yes, we can determine causation with this one. Typically with randomized experiments, the population's kind of hard because they're all volunteers. They're all students in some psych class for extra credit or some crazy thing like that. But this started out as a random sample. So I thought we'd go back and look at what the author said. So first of all, just on the strength of evidence, the author said to assess effectiveness of interventions in a population-based sample, a necessary step is to understand how the interventions are likely to affect the general population. So they're talking about this population-based sample. They also added, it's also unclear how our finding would generalize to other populations and communities, although there's no reason to conclude that Chinese elders would respond differently to other elders if similar interventions were done in Western populations. But additional work is needed to show that. So this is exactly the stuff I would be talking about in my class. It's like, people like those that were in the study, but it might go further than that. Still on this, now moving to the step 6, look back and look ahead. So the researchers thought that physical activity would help increase brain volume. So going into that. So they thought Tai Chi and the walking would be those 2 that would increase brain volume. They did not find that. They found the Tai Chi did, but they did not find the walking did. They found the social interaction did. Wow. But they did, however, find that the walkers who walked faster had improved scores on some cognitive tests. So they did something with their speed of walking that they were calculating as well. They thought the fast walkers did show some improvement on there. And they thought that those subjects kind of living in a big city like Shanghai probably did quite a bit of walking just as a function of daily life. And if they're going out to the track to walk on the track, they're really not doing anything differently than they've done day after day after day. But they thought if they move them and make them walk faster than they usually do, that might be beneficial. So they thought, kind of looking back, if they had to do this over, they'd have some sort of mechanism where they had to walk at a certain pace. And they had to keep this pace up that was increasing their heart rate more than it typically would in their daily life. And the authors thought that the level of mental improvement for the novice Tai Chi subjects was making the difference. So they didn't want people in the study that were already involved in Tai Chi. And if they thought somebody was very familiar with Tai Chi and they were doing Tai Chi with a bunch of beginners, it's got no effect because they're just daydreaming their way through that. And then, kind of like we saw, they said the magnitude of brain growth demonstrated was relatively small. So it wasn't this big difference, like, wow, what a huge difference it is. We got a p-value that was small, but not really small. And they thought a longer duration study with an increased sample size would be necessary to kind of really show there's some good potential interventions for reversing brain atrophic process and maintaining those cognitive abilities. Getting that oxygen flowing to the brain. All right, transitioning to theory-based methods. So the F statistic is what you use in ANOVA. And of course, that's the ratio of that between group variability to within group variability. Kind of find out how far things are apart on the top and how much spread in those groups you have on the bottom. And I don't have my students calculate this in any way, but I want them to understand what's going on. You know, like when you're doing just like a T statistic, the top is how far apart are your means from another. And the bottom is all the standard deviation sample size stuff in group within group variability. So it's very much the same. And then these are the validity conditions we use for a ANOVA test. You know, we say the F distribution is a good approximation for the null distribution of the F statistic. So we're going to see a null distribution using not just the mean group diff, but the F statistic as well. And that theory-based distribution will fit that nicely if either the sample size is at least 20, we had that in all our groups, without strong skewness or outliers, or if the sample sizes are less than 20, then you need some sort of symmetry to that to make things work out. And then the variability should be about the same in that. And what's typically used is the largest standard deviation should be not more than twice the smallest on that. So we're going to look at that, but we're also going to look at really what was our simulation-based result? Does that match up with the theory-based result? That also shows there could be a good fit or not. Not. So here's our data, again, broken up in those groups. So sample sizes, as I said, yeah, they're all more than 20. So we're good there. Do you have any strong skewedness? Yeah, it's skewedness, but I don't know that I'd call it strong. And then the standard deviations, there's a bit of a difference here. So the smallest is this 0.697. The largest is 0.387. But it's still just barely within that factor of 2. So I think we're fairly safe there. Oh, and then another thing I talk about after I get done with kind of with every test is talking about strength of evidence. I'm really talking about power here. So what makes for a powerful test? What gives us that strong evidence? So as sample size increases, that strength of evidence will increase. So when I went to my smaller sample sizes, you know, I got the bigger p-value, but when I went to the real data set, you know, I got the smaller p-value. And they would have seen this over and over and over again. And of course, as the means move farther apart, the strength of evidence increases. That's that numerator, you know, in the F statistic. But then the denominator, as the standard deviation increases, we get more noise in the data. That strength of evidence decreases. That's that variability within the groups. And kind of like Jill talked about last week, doing matched pairs tests, that's the way we decrease variability within the group so we can get strong evidence. But these are all exactly the same things we would have looked at when comparing two means. All right, so now I'm going to go back to the applet. So these are my points to try to remember as I do this. So we're going back to the applet. We're going to have the applet calculate the F statistic and see how we can do simulation with that. And then we're going to see how our simulated null distribution fits in with that theoretical null distribution and how those p-values line up or don't. We'll look at the follow-up tests, and then we'll take a look at the ANOVA table as well. Oops. And now I got to go back to the applet. So let me just kind of remember this p-value for our mean group diff statistic goes about 0.03. I'm going to unclick that so it's gone. And down here on the statistics, besides mean group diff, you've got a number of other things. So one of those is the F statistic, which I'm going to look at. So click on F statistic, none of this changes. It's all the same data, same mean, same standard deviation. But now here's our observed F statistic down here. So 3.109. So when I go up here to do my shuffles and I shuffle the responses like I did before, I get similar results here, but it's calculating a F statistic that could happen if the null were true. And it plots it up there, 0.39. And again, you can do that again and again and again. So here's 3 of them, and here's 997 more of them. So you get this shuffled distribution of F statistics. And if I put in the observed 3.109, right about in here, and I get a p-value, it's right around 0.03, just like we did with that mean group diff statistic. Could do more shuffles, see if it sort of stays there. It's like it's just above 0.03. And so again, this is still simulation. I'm just using a fancier statistic. And so now to do theory, just click on this overlay, the F distribution. And you can see that p-value is just below 0.03. So very similar. They all gave us very similar p-values. So this is the theory-based one. Seems to be working, seems to be fitting in with our simulation. And then you can also do all this down here on this ANOVA. You can click on the ANOVA table and it gives you output very similar to ANOVA. So you've got the sums of squares. Of course, that's where R squared is getting calculated. R squared, if I click on the R squared, there's that 0.03. And if I click on, just hold it right there, you can see the 10.83. So that's that sum of squares, 10.83. Then divided by the total sum of squares, the 130, that's the 0.083, 8.3%. And then the F statistic given here, p-value, again, just below 0.03. And then how it's calculated is with these two numbers. So that mean squared from the model, that's the variability between the groups. And the 1.16, that mean squared error, that's the variability within the groups. And then lastly on this slide, or this applet, here's the confidence intervals. So here's really the follow-up test we do. There are many kinds of follow-up tests. We're just doing just pairwise confidence intervals, all the ways we can pair them up. And if they have asterisks by them, that means the interval is either completely positive or completely negative. Zero's not in there, that there is a significant difference. And you can see Tai Chi is significantly better than walking or none. And then the social was significantly better than none. So I guess social and walking didn't quite make it there. That's why the other ones that were one's negative and one was positive. So just got above 0 on that one. All right, any applet questions before I go back to my last slide? All right. Oops, that button. Find it somewhere here. I have to start all over again. All the way to the end. There's my last slide. All right, sources of variation diagram. So this is simply something that Karen brought up a couple of weeks ago. And it's a way that we look at variability in the data, how the experiment or the study was set up. And I sort of put it here at the end because, oh, I thought I'd add it. It's not part of the module. But then I sort of liked it at the end because I thought it gave a nice summary of kind of what we did in here. You know, we're observing variation in the brain volume change over those 40 weeks. We had a whole bunch of inclusion criteria, you know, residents of a specific neighborhood in Shanghai, that age range, healthiness, ability to walk, all those sorts of things. Yeah, design. I could have added a lot more on the design things here. So all programs were 40 weeks, but there are other things, you know, the times was all the same. They're trying to make things very much the same in those groups, except for the activity on there. And then the sources of explained variation, well, that's that activity type. That's what we looked at. The Tai Chi, the walking, social, and control. Save the walking speed here for last. And then sources of unexplained variation. So you get your 4 groups, you randomly assign people to these 4 groups. You want those groups to be very similar, but there's still variability within all those groups. You know, the activity of the subjects. So some subjects are probably very active naturally. And maybe when they go to some intervention, they go, well, it didn't really change things. That's what I normally do. But if a subject's not that active, that intervention might make a huge difference. The health of the subject, same sort of thing. The mental ability of the subjects. You know, they wanted them not to have dementia, but there's still variability in the ones they have left. How well they were engaged in activity. We saw that with the walking speed. Some walk slow, some walk fast. Maybe some Tai Chi people were just totally into it and some weren't. Maybe in that social interaction, some were really involved and some just sat in the back. And then here I'm sort of making things up. I have no idea. Total brain volume. People have different brain volumes. Does that affect sort of that change in brain volume over time? MRI accuracy. I'm assuming they're not perfect. That you go one day, you get a different number than you'd have another day. And then the walking speed. So I put that both places. So it's sort of a sources of unexplained variation because they did not include that in the numbers we saw, but they did include it somehow. They noted on some cognitive test the people that walked better showed improvement. So it's sort of explained and unexplained. Kind of put that in both places. All right, that's the end of my slides. Now, if we got time for some questions.
Thanks very much, Todd. We do have a few minutes till the top of the hour, so feel free to type a question in the chat or unmute and ask a question if you'd like. Any questions for Todd?
I did have a question for you. I just did this unit a couple of days ago, and I was aware I had read, you know, of course, Wikipedia is a great source for getting things you don't know. And so then I looked at Wikipedia about if you take the value of alpha and you divide it by the number of comparisons that you do, that you can use that, and just not that I would present that to the class to have them do it, but just as a thing that I would say we could even do this and that would take care of some of that problem, just as a thing to mention, right? Not to go into great detail with it.
Right. Yeah. Doing those follow-up tests, there's a number of ways to do that. There's Jill might help me remember it. It's somebody's name, right? With that dividing.
Bonferroni was
Bonferroni.
Anyway.
See, I'm retired. All that stuff is flying out of my head. Bonferroni correction.
Yeah, I do it in more advanced stats class, which we call 219 now. It has more additional topics compared to introductory statistics, right? I mentioned word Banferroni just because it sounds exotic, but never explained it in the introductory class, but you're right. Mhm.
Yeah. In the intro class, you're always walking that fine line of how much to tell them and how much not to tell them. Because if you tell them too much, it just takes away from what's really important. They can only handle so much. So if you're throwing something else in there, something's got to come out, I think, sometimes. So you got to highlight, you got to beat on those important things over and over and over again.
Time for another question or two. Any further questions, comments, reactions? I think I'll stop the recording here.